You're listening to KTAL LP FM 101.5 in Las Cruces and streamed live at radioketal.org, that's Quetal, and also at lccommunityradio.org. And there you could also find the place for donating. If you'd like to become a member, we'd really appreciate it. Right now, you're listening to Science. Let's take a look. This is every Tuesday, 12 to 1. I'm your host, Vince Gucic, and we go over all diverse fields of science, engineering, math, and I often have guests who are working scientists and engineers. With us today is Professor Kathryn Hanley from the Biology Department at NMSU. She is an international researcher and recent past chair of the American Committee on Arthropod-Borne Disease Viruses. And those are those worrisome agents that you've heard about, like zika, dengue, chikungunya, and I will let her tell everybody how she got into this and what she's doing.
Thanks, Vince. I have been working on arthropod-borne viruses, primarily mosquito-borne viruses, for the last 20 years. So my initial training was in the evolution and ecology of host pathogen interactions. For many years, I worked on lizards and their parasites, but I developed virus envy because I was just amazed by the experiments that people could do with viruses and also how much their work contributed to improving public health. So many years ago, I took a position as a research associate at the National Institutes of Health, and there I began developing, along with a large team of people, a vaccine for dengue virus. And as we were developing that vaccine, and as things were going better than vaccine development usually goes, and so things were looking very promising, because I'm fundamentally an evolutionary ecologist, I started to worry. So things were going too well, so I started to worry. And I thought, well, what if this vaccine really works well and we're able to eradicate dengue from humans? How did dengue get to humans in the first place? This virus didn't originate with humans. So in fact, we knew at the time that it originated in what we call a sylvatic cycle, that is a cycle in non-human animals, and that sylvatic cycle occurred in the canopy of forests in Southeast Asia and in Africa. And so if that ancestor is still out there, could it return to infect humans? Could it reestablish dengue? Could there be strains in that sylvatic cycle that the vaccine doesn't cover, and so we're actually opening the door to the emergence of something even worse? And that's
I remember,
really,
you gave a really interesting talk at NMSU in the chemistry building, and you talked about bridge vectors, and that's the way it could jump to other places?
Exactly. So what we're thinking about here is a virus that's probably maintained in monkeys and other species up in the forest canopy, but of course people don't generally climb up into the forest canopy very often. However, there are mosquitoes that are a little more, we like to say, promiscuous in their feeding habits, and so they will move up into the canopy to feed on monkeys, but then if humans move into the forest or if humans cut the forest down, those mosquitoes will also willingly bite people. And so you can then have the transfer of viruses from a host that would normally be very restricted from access to humans and into humans, and thenceward onto transmission through the world.
Is there anything about the genetics of what makes a virus able to transfer between primates? Like, we're reasonably different.
Right. So when you look at the big emerging viral threats, and not just those transmitted by mosquitoes, but when you consider dengue, chikungunya, zika, influenza, even go to human human immunodeficiency virus, which emerged from simian immunodeficiency virus, they all have a genome that's made up of RNA. And so for people for whom that's just 3 letters, human beings have a genome that's made up of DNA. And DNA is pretty stable. It doesn't change very quickly. RNA, on the other hand, mutates about 1 million times faster than DNA. So it's very genetically labile, and we think this contributes to the ability to rapidly adapt to new hosts and establish new transmission cycles.
Okay, so that covered what I wanted to ask you about the nature of the viruses. Maybe you could go into some more detail about how the virus actually gets into a cell.
So I'd love to be able to tell you that in detail about dengue virus, but frankly, we don't know. So all viruses have to bind to what's called a host cell receptor. And that's a little bit of a misnomer because that's going to be a molecule on the host cell that has some function that benefits the host cell. But the virus has then evolved to utilize that molecule and use it like a lock and key system to get into the cells where it's able to replicate. So for something like influenza virus, we have a very good idea of exactly what the receptor is at sialic acid, exactly what the protein is on the surface of the virion that binds to sialic acid and lets flu in. For dengue and for West Nile and for yellow fever virus, nobody has ever been able to identify the specific receptor that's necessary for entry. In fact, there may be a number of different molecules on the cell that lets the virus in.
That's interesting. How long have these viral diseases been studied without success in that regard?
Right. So virology is one of the youngest fields of science. So we didn't actually have a working idea of what a virus was until the very late 1800s. We didn't know that there were viruses of humans until the early 1900s. And part of the reason for that is that viruses are smaller than the wavelengths that comprise visible light. So that's about 400 to 700 nanometers. Viruses are smaller than that. So bacteria are bigger, and when we put bacteria under a microscope, we can see them. You can put viruses under a standard light microscope, you will not see them. And so we couldn't see, quote unquote, see viruses until the development of electron microscopy in the 30s, 40s, and 50s. And then we began to be able to actually visualize viruses. And shortly thereafter, the technique of cell culture was also developed. So people suddenly realized that you could take cells, put them in a plate, give them the right nutrients, and they would grow. Before that, if you wanted to culture, say, yellow fever virus, you had to do it in a monkey. And that was extremely difficult and, frankly, extremely dangerous. Many early researchers in virology died, not only from the viruses that they themselves were studying, but because monkeys are infected with a herpes virus called monkey's B virus that's fatal to humans.
Really? I'd never heard of that.
Not all monkeys, but macaque monkeys, which are the most standard laboratory model, carry herpes B. And so if you're bitten by those monkeys, nowadays you go on a lifetime course of antivirals, but in the 1930s and 40s, you died.
A lifetime course?
Yes. So you really don't want to develop herpes B virus. Anyway, once we had plates and we could not use monkeys to any more than was strictly necessary, that really advanced the study of viruses. And then, of course, in the 80s, 90s, and now, we have the advent of new sequencing technologies that have opened the door to discovery of viruses that we can't grow in an animal and we can't grow on a plate, but we can find their genomes, and now we can begin to characterize them.
So I remember you said you were also looking for a small animal model. How does that fit in?
So we can do a lot with cells on a plate, but one thing we can't do is easily test safety or efficacy in a full living model of drugs or vaccines. And when Zika virus first became prominent in 2015, it was immediately appreciated that we really, really needed drugs to treat particularly pregnant women to protect their babies from the terrible birth defects conveyed by Zika infection. We'd also love to have a vaccine to protect people against Zika. And so it's easy, it's not easy, but it's straightforward to do the initial formulation of a vaccine, the initial formulation of a drug. But you need a living animal in which to test the safety and efficacy of that drug. And so we needed small animals that could be infected by Zika virus. The difficulty being Zika doesn't infect wild-type mice, and mice are the go-to model. Zika doesn't infect normal rabbits. And so we had to tinker with these mice. We had to shut down components of their immune system in order to finally create a mouse in which Zika replicates nicely and causes some of the manifestations of disease that we see in humans. And
how would you know what to shut down?
That is always an educated guess, right? It's a hypothesis, but In our case, we know that the front line of antiviral defense is the interferon response. That's what is the first response, really, that's marshaled by the immune system against a viral infection. So we utilized mice that were deficient in components of the interferon response, and in fact, they're quite susceptible to Zika.
Uh-huh. So how is that progressing?
That model has been very useful. People have also elaborated upon that model to make it even more human-like. So you can engraft mice with human immune systems, for example. You can engraft mice with human livers. You can do an extraordinary amount of manipulation to mice to make it a more and more realistic model.
So it's well out of beta testing, you might say.
Oh, it's well out, yes.
Oh, okay.
The other work we've been doing, though, is that the main concern with Zika virus is microcephaly, the small head,
right,
that appears in about 11% of children born to women who are infected within the first trimester of their pregnancy. So we've also been working with researchers at University of Washington and elsewhere to try to develop a macaque-monkey system wherein we could infect the monkeys and see recreation of some of those manifestations in the fetus, and then ask whether we can use that as a model to test protective drugs or protective vaccines.
So then you've got to get herpes B free.
Right.
Macaques.
Well, we've gotten much better at handling macaques so that you'd never actually have to really come in contact with them. So there's telemetry systems where we can monitor their temperature without ever touching them. They're happier. We're happier. The less we touch them, the happier we are. Yes.
Okay. So I just read somewhere, maybe Nature or Science, about the microcephaly abating, and this article attributed it to something like herd immunity, that so many people have been exposed that they're not even seeing the symptoms. Is that a realistic view?
I have no doubt that herd immunity contributes. So we're certainly seeing a lot less infection and microcephaly this year than we did the previous year. And that is, in many ways, the signature of a typical epidemic. There's a huge number of infections. Science and scientists rush toward interventions. But even as the interventions are coming online, very often the number of infections is declining because the virus or the pathogen has burned through most of the population. And what I expect we're going to see is that Zika will settle into something like chikungunya or dengue in the Americas, which is that it's always here. And sometimes it's more common and sometimes it's less common.
Speaking of always here, what's always here in the U.S.?
Right. So I guess there's a couple answers to that question. One is that we know that there have been small outbreaks of dengue virus and chikungunya virus and Zika virus in the southern U.S. recently. So we know, let me even back up and say, dengue was first described in Philadelphia. So the vector for dengue, the Aedes aegypti mosquito, used to get as far north as Philadelphia and New York in the summers. It would then disappear over the winters and be reintroduced to port cities. And while it was there, you got active transmission.
You mean, does it have to be mediated by the mosquito, or it can be person to person?
So the only of the viruses that I'm interested in, we are not aware of sexual transmission of dengue or of chikungunya virus. There is documented sexual transmission and perhaps other routes of transmission of Zika, but they are still very, very uncommon compared to mosquito transmission. So really what you worry about is mosquito transmission.
Okay, very good. I'm going to take a short break here, and then we'll get back to you. We're talking with Professor Kathryn Hanley from biology at NMSU about some of the interesting emerging viral diseases